Thank you all for joining us. So today we have our CRESP seminar and Steve Kustka is going to present it. His presentation is titled Investigating Radiosynthesis, a Potential Metabolic Pathway in Melanized Fungi. A little about Steve. So he's a radiation health physics PhD candidate at Oregon State University, working alongside Dr. Catherine Higley. Following the completion of his BS in physics from Rowan University, New Jersey, Steve began his career as an RHP technician at the University of Pennsylvania in 2016. During this time, he began the Oregon State RHP e-campus program in 2018, prior to relocating for his PhD program in 2021. He is currently the CRESP Early Career Professional Committee co-chair. His dissertation research project consists of acquiring additional evidence to support or refute a hypothetical metabolic pathway utilized by melanin pigmented fungi capable of converting ionizing radiation into cellular energy through so-called radiosynthesis. Collection of additional supporting evidence has implications for bioremediation of nuclear waste sites and risk mitigation of cosmic radiation in low Earth orbit and beyond. So with that introduction, the floor is all yours, Steve. Take it away.
Thank you so much for Panjali, and thank you all for tuning in. Today's presentation is going to be a little bit different from our typical seminar in the sense that this presentation is intended to satisfy my prelim exam. Prelim exam is where I produce 3 peer-reviewed, I'm a little bit ahead of myself. The prelim is where I propose 3 experiments to my wonderful graduate committee in order to produce 3 peer-reviewed publications that are intended to fill gaps in the larger scientific body. We'll start off with a little bit of a roadmap of what to expect. We'll begin with some unit and terminology preference, and then dive briefly into the literature review, or as I like to think of it, the lore behind the topic, followed by our proposed experiments and some of the relevance behind this topic. We'll start with the only equation in this presentation, effective radiation dose. It's basically the summation of absorbed dose from all individual interacting types of radiation within a specific organ. We usually use this to describe whole body dose, especially in terms of radiation biology. There's quite a few biology terms here, but I will include them in the applicable slides within the footer. NADH and FADH2 are enzymes, or catalysts for biochemical reactions that are often associated with metabolism. NADPH is a coenzyme of NADH, and it's associated with anabolic reactions, such as DNA, protein, and cell membrane synthesis. NADH and NADPH are also coenzymes, and they can be flipped by a single enzymatic reaction. And finally, we have ATP, or an energy currency that is responsible for driving cellular function. Health physics and biology have some overlapping terms. For instance, radiolysis, which is a chemical interaction where radiation splits water molecules. The byproduct of this is often reactive oxygen species, which are highly reactive, and they tend to strip electrons from stable atoms or molecules, thus breaking them. And they're also naturally produced in normal cell function. 3.6 Rankine, not great, not terrible, and life finds a way. On April 1st, 1986, the world experienced the catastrophic failure of the Chernobyl nuclear power plant, releasing an entire reactor inventory worth of nuclear fuel and fission byproducts directly into the biosphere. Between 1987 and 2001, exploratory missions uncovered the presence of 2,000 strains of 180 different fungal species, both within the exclusion zone and perhaps more surprisingly within the reactor unit itself. Large concentrations of oxygen-respiring melanized fungi were discovered to be actively growing towards ionizing radiation sources and even consuming hot particles, including the graphite neutron control rods. Later publications experimentally confirmed increased rates of growth and spore germination relative to their counterparts at lower ionizing radiation levels. The culmination of these two papers coined the term positive radiotropism, where fungi were beneficially interacting with radiation. Three of the most abundant species included the naturally melanized yeasts Cryptococcus neoformans and Vongelia dermatitis, as well as the extremophile hyphae-producing Cladosporium sphaerospermum, all of which were found to thrive within the devastated reactor. The abundance of these three species within the reactor were largely attributed to the melanin pigment. Eumelanin, henceforth melanin, is a dark biological pigment found in humans, fungus, bacteria, and is the primary component in squid ink. It is produced via the polyketide synthase pathway through the oxidation of tyrosine and is even metabolized under conditions of starvation. Melanin is responsible for very robust survival capabilities. It's capable of dissipating 99.9% of all excess excitation energy as heat, and is believed to reduce the pore size to prevent water loss similar to some plants. It also protects against external redox agents such as bleach and permanganate, offering significant protection against chemical lysing. Deposition also is largely dependent on an organism by organism basis. Synthesis can occur on the outer, inner, or intermembrane, and even the cytoplasm, particularly within bacteria. Melanin is constructed almost entirely of stacked pi bonds. This allows electrons to flow freely throughout the entire molecule without severing it. Because of this, it can exist in a reduced, oxidized, or neutral state, allowing melanin to function as a stable free radical. Also, perhaps most surprisingly, depending on the concentration, melanin has electron transfer properties that resemble those of semiconductors. The unusual protection and electron characteristics of melanin laid the foundation for so-called radiosynthesis. The team behind the 2007 flagship publication claimed that radiosynthesis functioned very similar to photosynthesis, in the sense that it was pigment-mediated. Two further studies, a study claiming lower ATP concentrations in melanized fungi relative to their non-melanized counterparts, could be attributed to some form of carbon fixation. And a genomic study of a fungus tested in the initial manuscript suggested that ionizing radiation was being harnessed for protein biosynthesis. Due to the high molecular weight, density, and electron characteristics of melanin, it was hypothesized that melanin was capable of capturing Compton-scattered electrons and low-energy scattered photons for metabolic purposes. Additionally, reactive oxygen species were also hypothesized as being quenched or nullified by melanin, with electrons gained from this interaction also used for metabolic purposes. In order to understand how these captured electrons and photons could be used for metabolism, it would be a pretty good idea to cover traditional metabolic pathways. We'll start with covering photosynthesis, largely due to the analogous nature between the two, and then move into aerobic respiration, which is a metabolic process utilized by both photoautotrophs, such as plants, and saprotrophic organisms, such as humans and fungi. Photosynthesis begins with the light-dependent reaction, where photons of about 2 to 3 electron volts are absorbed by chlorophyll pigment in a protein structure called photosystem II. Photons then excite the electrons, typically from an S0 to an S4 state, and these excited electrons are then shuttled by the electron acceptor phaophytin into the photosynthesis electron transport chain. As the electrons move through these protein complexes, an electrochemical gradient is established, and this gradient causes protons to flow through the ATP synthase, the structure all the way on the right, which is ultimately responsible for synthesizing ATP. In order to fill the electron hole now present in photosystem II, water molecules are oxidized by charge separation in the oxygen evolving complex, the only known protein complex of its kind, to split two water molecules into their respective constituents. The light-independent reaction of photosynthesis is essentially why we love photosynthesizing organisms. Through a series of stepwise redox reactions, the protein rubisco sequesters CO2 from the atmosphere and converts it into glucose. When photon capture is inadequate to drive the Calvin cycle, glucose stocks are utilized in a process shared by many organisms. But this, of course, is aerobic respiration. Aerobic respiration begins with glycolysis, where the glucose is broken down into pyruvate, which is a metabolic intermediate used in later respiration steps. 2 NADPH and a net yield of 2 ATP are also produced. Pyruvate is then shuttled to the mitochondria, where it undergoes the Krebs cycle, or citric acid cycle. Here, pyruvate is decarboxylized, a CO2 molecule is replaced, and protonated with a hydrogen atom, transforming it into acetyl coenzyme A. The citric acid cycle ends with the production of the reductants NADH and FADH2, reductants being electron carriers, and GTP, a building block for ribonucleic acid synthesis. Oxbows produces the bulk of ATP in aerobic respiration. The citric acid cycle combined with oxbows produce a theoretical max of about 34 ATP, but this is actually closer to 30 or 32 due to electron leakage, which is the cause for natural ROS generation as a respiration byproduct. A quick fun fact, from the start of glycolysis to biosynthesis of ATP through oxbows, about 2,900 kilojoules of energy is released. This is the potential energy equivalent to a little under a gram of black powder, so it's a pretty good thing this happens over time. Returning, though, to our original question, can we say that photosynthesis and radiosynthesis are analogous? The answer is not quite. We understand the photosynthesis mechanisms very well. Photons come in, excite chlorophyll, the excited electrons are handed to thayophytin, which ultimately produce NADH and NADPH. Whereas with radiosynthesis, it's much more complex. Photons of much higher energy, several orders of magnitude higher than that of visible light, are capable of ionizing melanin and ionizing water to produce radiolysis byproducts of ROS. The interaction of electrons between melanin and ROS is something of a difficult thing to describe, and ultimately, there's an unknown electron transfer mechanism that must be possible for the electrons in melanin to produce NADH and NADPH. Some of the strongest claims for radiosynthesis in Dadashova et al. Come from growth assays, insinuating that radiation increases colony-forming unit counts and growth rates, in addition to dry weight biomass relative to their respective control groups. One such experiment was performed using naturally melanin-producing Cryptococcus neoformans, and a mutant strain that is incapable of producing melanin without the incorporation of a precursor, or L-DOPA. This experiment ultimately led to the conclusion that carbon was being fixated 3 times faster in melanized counterparts between irradiated and unirradiated controls. I want to take a quick step back, however, and kind of insinuate if it was the carbon substrate or the radiation that was capable of sustaining such a cell, or cell growth as evident in the system. Making a few assumptions, which health physicists love to do, we can roughly calculate the binding energy of each neoformin cell. Dividing that binding energy by the amount of energy that was deposited in the system, according to Dadashova et al., this nets us around 115 cells. While not appropriate, since radiation energy deposition is intended for metabolic purposes, we can determine that the amount of acetate present in the system is ideally the driving factor behind the amount of cells that were present, and that the theoretical maximum energy deposition from the radiation is incapable of sustaining a system this large. There are some experimental issues as well. The unirradiated melanized cells exhibited a significant decrease in counts and uptake relative to their unmelanized counterparts. This could be explained by melanin reducing the uptake rate of nutrients and carbon sources, such as how it reduces water loss. Additionally, the incorporation of L-DOPA affects metabolism and growth rate baselines, also leading to a longer lag phase following irradiation. And perhaps most importantly, the experiment showed an increased substrate use was present, but it failed to address exactly what the radiolabeled carbon was used for. For instance, the non-melanized samples did display higher carbon uptake, but it fails to mention if this carbon was used for higher repair or for metabolism's sake, or biosynthesis. Another growth experiment was performed using naturally melanized and artificially demelanized samples under sucrose-rich and sucrose-deficient conditions. Samples were demelanized using the fungicide tricyclazole to inhibit melanin synthesis. The results were interpreted as melanized cells in substrate-rich agar plates grew faster and larger than their non-melanized and substrate-lacking counterparts. Again, there are a few inconsistencies with this. Artificially demelanizing control samples with fungicide introduces a confounding variable by directly affecting metabolic and growth baselines. This was confirmed in a paper by Robertson et al., which initially supported claims of radiosynthesis. Several inconsistencies also arise from the data presented, or rather the lack thereof. The initial paper claimed that growth measurements were taken for every day for 15 days, but only 4 data points are shown. Additionally, the unirradiated, melanized carbon-starved and carbon-rich samples exhibited similar growth rates, indicating the presence of some possible confounding carbon substrate or even enzymatic breakdown of the agar. Finally, a radiation of melanized and non-melanized groups exhibited similar increases in growth, echoing that there may be an alternative mechanism other than melanin responsible for driving growth rates. Another key piece of evidence lies in the use of the tetrazolium salts, XTT and MTT, to determine NADH concentrations within the cell. Negatively charged XTT remains outside the cell, while positively charged MTT is intended to permeate through the cell membrane. Because the reaction is typically slow, or I should say the XTT redox reaction is slow, ferrocyanide is typically added to expedite the reaction rate, making it realistic. The results appear to indicate a 4-fold increase in NADH oxidation, and thus a 4-fold increase in NADH present in the cell following 40 minutes of irradiation. However, the initial NADH and ferrocyanide redox velocity is fairly equal among all treatments. This finding, supported with those in an article by Bryan et al. 2011, indicate that ionizing radiation may not be the only treatment capable of increasing metabolic stocks. There are also 2 issues with the experimental methods. A fairly recent publication has shown that MTT is incapable of permeating melanin cell walls. And additionally, the salts can be reduced by melanin itself or superoxide, which is a reactive oxygen species that is abundantly produced by radiolysis. This means that melanin may be solely responsible for the reduction of XTT and MTT, therefore giving us an inaccurate representation of metabolic stocks. Overall, one could claim that Dadashova et al. Fails to support the use of radiation for metabolism. Rather, it supports the idea of positive radiotropism. There's also a large amount of inconsistencies, both in methods and data interpretation. And I'd also like to point out that there is a very small pool of radiosynthesis data in general throughout existing literature, as well as a very small number of independent research groups. Failure is always an option. Our 3 experimental objectives are to fill gaps pertaining to the use of carbon substrate, fill gaps pertaining to ATP concentration and possibly the reductants NADH and NADPH, and also to perform an RNA sequencing differential. Now, I admit I'm not a classically trained microbiologist, so I have to follow the KISS method. And no, not the rock all night and party every day method, but rather keeping it scientifically simple. The perfect experimental candidate is the naturally melanin-producing Cladosporium sphaerospermum, specifically strain CBS2. It is slow-growing, allowing us to commit to well-timed experimental procedures, and is extremely resistant to radiation and other environmental stressors, evident by it being the most abundant species within the Chernobyl nuclear power plant. The organism is also under investigation for a host of purposes, making it an ideal candidate for radiosynthesis experimentation and expanding upon the greater body of science in general. For example, It has no fully mapped RNA transcriptome yet. For the next couple of slides, I'll be including a flow diagram on the side for a visual reference. All samples will be naturally melanized to avoid confounding factors such as L-DOPA and tricyclism. Samples will be incubated on Saboron dextrose agar at 25 degrees Celsius, as that is the medium for the most robust growth. We'll also be incorporating a 10 micrometer pore filter on top of the agar, as this will allow hyphae to penetrate into the agar and collect nutrients from the agar, while simplifying sample extraction and minimizing agar cross-contamination. We also want carbon to be the limiting ingredient in the cultures, while allowing sufficient nitrogen and phosphorus content as they're needed for protein and ATP biosynthesis, respectively. To investigate growth trends, we will have to perform our experimental procedures in both the stationary and exponential growth phase. This is because different phases can have drastically different physiological responses to stressors. To ensure that we can reach the stationary phase in a realistic time, ideally less than 2 weeks, carbon stocks will be reduced from 40 grams per liter to 10 and even 4 in order to expedite carbon starvation. Growth will be determined by 2 caliper measurements twice a day, ideally one in the morning and one mid to late afternoon. One measurement will be longitudinally and the other one will be latitudinally, and these measurements will be averaged and plotted to construct our curve. Part of our experimental process involves doping samples with astaxanthin, a very powerful antioxidant pre-irradiation, and in control instances in case there is an introduction of a confounding factor. Another fun fact, astaxanthin is what gives shellfish their distinctive pink color, which also gives my friend Jeff over my shoulder his distinctive color too. The purpose of adding an antioxidant is multifaceted. We want to allow electrons to flow as freely as possible through melanin, especially since melanin is unable to affect intermembrane reactive oxygen species. Astaxanthin will also reduce the frequency of ROS, which will reduce biological damage from irradiation. This may also possibly offer some insight into the magnitude that ROS signaling can have on the cell, as this may have a significant, or may play a significant part in cell growth and metabolism. Per our irradiation conditions, samples will be irradiated to 100, 500, and 1,000 gray. We want to use such high absorbed doses to account for the incredibly low probability of ionizing radiation interacting with cells at low doses. High dose rates also ensure uniform irradiation of the medium, allowing for reasonable amounts of energy that may possibly be used for metabolism to be absorbed by the medium, and to also allow plenty of opportunity for the melanin pigment to harness these interactions. In the chart in the middle, you can see the amount of cells that are possible from the Dadachova experiment, as well as the amount in acetate and the amount of acetate used per milliliter. The top is the radiation center irradiation system, or cobalt-60 irradiator, I should say. And I have that value in there to give you an idea of how many cells are theoretically possible from the energy that will be deposited. While it's not the same as the amount of acetate that you would expect from a gram, it's pretty similar, and it may have some interesting results. I'm including a chart to give you an idea of how we will be making our comparisons, because we're expecting a lot of data, which is desperately needed. For instance, we'll be comparing treatments within each group. In the stationary phase, under the antioxidant conditions, we'll compare background to the expected treatments. This will occur in all of our other groups. We'll also be comparing between antioxidant and no antioxidant treatments within both phases, and then we'll be comparing groups cross-phase. Before we address our experimental methods, I do want to discuss how cells utilize carbon. Carbon is used for 3 main purposes: DNA and cellular damage repair, biosynthesis and polymerization of new cells, and processes linked to central metabolism. Typically, cells prioritize cell maintenance and central metabolism over biomass production, and they only undergo cellular growth after non-growth requirements are satisfied. Adding an antioxidant reduces the need for cell maintenance and it may offer insight into whether ionizing radiation is harnessed directly into metabolic energy, thus allowing for C to be funneled directly towards biomass production. Our first experiment is intended to address carbon for growth versus metabolism and repair, and to add evidence to support or refute a possible carbon fixation method similar to the light-independent reaction in photosynthesis. This will be determined by two means. First, we'll be quantifying if growth over time occurs in the stationary phase, or if samples exhibit increased growth rates in the exponential phase that exceed the amount of growth permissible by the carbon substrate. In order to determine carbon fixation, we will determine the percent mass of carbon in the sample via combustion analysis. CHN analysis requires some sample processing. Samples will have to be removed from the mesh and then centrifuged for 5,000 times gravity for about 20 to 30 minutes to ensure all fungal mass is collected and minimize the presence of agar. A fungal pellet will then be loaded into a glass filter, weighed, and undergo combustion to separate chemical constituents via gas chromatography and quantify the particles present. To quantify internal ATP concentration and possibly NADH and NADPH, we will utilize the well-vetted commercial luciferin-based ATP assay. There are similar assays for reductants as well. The reaction consists of the enzyme luciferase, generating light by the oxidation of luciferin, which is derived from fireflies of all things, and requires the introduction of an ATPase inhibitor to prevent ATP degradation following cell lysing. This reaction releases a peak emission of 560 nanometers, and it can be quantified with a plate reader. Typically, traditional ATP assay kits require cells to be enzymatically lysed, but this would cause issues trying to crack open the hard nut that is a melanized cell wall. Fortunately, we can utilize methods described by a team here at Oregon State University, which utilizes a series of sonication bursts on liquid nitrogen snap-frozen samples to physically lyse the cell. This method is particularly attractive since the stacked pi bonds associated with melanin, while they are very strong from a chemical standpoint, are fairly weak from a physical standpoint in terms of bond strength relative to their counterparts. We can confirm our lysing method to completely lyse the cells by attempting to reculture them following the sonication cycles described in Jones et al. Additionally, we may have to use the use of a picomolar concentration ATP assay kit. And this is to ensure that we have some detection as ATP turnover may be high in order to address various cell uses. And in the event that we cannot detect ATP, even at the picomolar level, it may be wise to then perform the reductant assays to establish which coenzymes and thus which cell processes are in action. Our final experiment lies with a RNA sequencing differential, and this is to determine exactly how the cell is responding. Since we can't talk to cells, we have to kind of dig into them a little bit more. The difference between DNA and RNA is that DNA essentially describes what the organism is, whereas RNA describes what is the cell doing, how is it responding. RNA is essentially a snapshot of a specific DNA base pair sequence, and this snapshot allows us to determine which genes are turned on or off, as well as which genes are up and down regulated. The RNA transcriptome is basically a recipe of RNA base pairs, but we have to convert binary to text. Since there is no existing library for CBS2 yet, we will have to make one. Fortunately, we can utilize the fully mapped transcriptome of the medically invasive UM834 strain to produce a hybridized de novo library. Again, we will need to construct a library in the stationary and exponential phase, as gene expression can also be incredibly varied between the two. And these libraries will not have an antioxidant incorporated into them, since we want to mimic conditions as closely as possible to that of nature. RNA sequencing differentials are exploratory by design, but several functions we want to explore include metabolism, cell growth, repair, nutrient transportation, and even ROS and ROS signaling. Great. So how is this relevant to CRESP and the scientific community at large? Well, you ain't done cooking until you're done cleaning, and space, the final frontier. We're all very familiar with the Hanford site. It's a absolutely gargantuan remediation task. This is largely due to there being about 54 million gallons of chemical and radioactive waste, in addition to 375 to 555,000 tons of uranium, plutonium production chemicals, and byproducts. Much of this contamination exists in the form of groundwater plumes containing large concentrations of tritium, anionic and long-lived radionuclides, and toxic metals and hazardous organic compounds. The use of a radiotrophic and possibly radiation metabolizing fungus has significant implications for Hanford and other nuclear waste sites in terms of bioremediation. Through the assimilation or degradation of nitrates and nitric acid, aliphatic and polycyclic aromatic hydrocarbons, toluene, and chlorophenols, we may have a very interesting technique for remediation. Interestingly enough, previous studies have confirmed that cladosporium can survive on toluene as its sole carbon source alone. Fungi are also very effective at adsorbing inorganics and reducing pesky metals such as hexavalent chromium. And of course, they're second to none at sequestering radionuclides. Fungi are also incredibly robust and diverse within the environment. They're able to survive in environments with low water content, highly acidic conditions ranging down to about a pH of 2, and can survive in high metal and radionuclide concentrations, making them an attractive alternative to large-scale, difficult remediation products. This is especially true since fungi can cover very large surface areas, and they're present in soils and wastewaters, and they can even penetrate through challenging matrices. Ultimately, however, there is a lack of literature despite a very high potential. I'd like to ask a question to the group. That is, what is the purpose of remediation? Risk reduction, right? When we think of risk reduction, we typically associate it with families, the biosphere, and members of the public. But there's been a growing interest for risk reduction of folks in a very unusual environment, outer space. As NASA and various other space agencies have big plans for the colonization of the moon and even Mars in the not-so-distant future. However, the largest threat for space exploration comes in the form of ionizing radiation. I've produced a little chart here to give you the idea of some of the effective doses that are expected. The average citizen of the United States receives about 620 millirem annually from natural and medical sources. This limit balloons in low Earth orbit of up to 18,000 millirem, which climbs even further to 38,000 on the lunar surface. And on Mars, because it has an atmosphere, very finite atmosphere, we can expect that to be about 26,000. These doses are greater than the known risk increase associated with ionizing radiation at about 10,000 millirem. And on the journey from Earth to Mars, which is expected to take about 900 days, some of the estimates range as high as 120,000 millirem. Fortunately, there may be hope. In 2022, a team published a low Earth orbit study where over the span of 622 hours, they deduced that radiotrophic fungi may be capable of becoming and utilized as a self-replicating bioshield. Over the time span, samples grew on average of 1.7 millimeters thick. This attenuated 0.84% more radiation across a 180-degree plane of protection. And in a 360-degree environment, this is expected to double. Cultures also grew 21% faster than their Earthside controls, which is very interesting. Finally, I would like to thank CRESP truly, I wouldn't be here without you, and I am incredibly grateful for this opportunity. I'd also like to thank Dr. Deb Deloche and Amy Carter of OSU Biosafety for helping me establish a biosafety level 2 lab in the RAD Center from the ground up. And also Dr. Scott Menn of the OSU RAD Center for letting me bring fungus into the radiation center. For science, of course. I have some references, and then some more references, and with my song and dance out of the way, I'd like to open the floor for questions.
Well, thank you, Steve, for such an amazing talk.
Thank you.
So let me start off the question and answers. So when you were going through the presentation, I know you were talking about space and colonization. So I'm just curious, like, what implications do you think that the radiosynthesis has on maybe astrobiology or like search for extraterrestrial life? Like, could the melanin pigmented fungi provide clues about potential life forms on other planets which are capable of harnessing the radiation itself as an energy source?
Sure. On the western coast of Australia, some of the fossil samples allegedly have melanin granules in them. And on the deep sea floor, life is believed to have come from hydrothermal vents. Several billion years ago, when ionizing radiation fluxes were higher before the natural decay of long-lived radionuclides like uranium, there may be some implication for that to occur, but it could also be that melanin was produced by these organisms for the sake of protection. So it might be, it's a really interesting concept to consider, but there's a lot more to go with it.
I can imagine. And just like another thing, so we're talking about Hanford, and like some of it waste we know is like pretty alkaline. Like I may have missed this, but do you think it's still capable of like, does it have an effect of pH? Like, do you expect it to have like lower efficiency at higher pH or something? I remember somewhere you mentioned a pH as low as like 2, I think.
Fungi typically like acidic environments, but they are capable of surviving a very broad range of pH levels. So they can survive, I want to say what I've seen off the top of my head is levels between 2 to 12, but it is largely dependent on species. And the pH also has an effect on which enzymes are produced, which play a key factor behind bioremediation.
Okay, thank you. I see Steven has his hand up. Go ahead, Steven.
Yes, thank you for your talk, Steve. I thought it was great. I have a couple quick questions. The first is about the experiments that you proposed. Do you know what like kind of time scale you expect the experiments to go on for? I know you mentioned like measuring like in the mornings and afternoons with like the calipers, but do you know like how long you plan for those to go for, I guess?
That's kind of difficult to say. Growth curve, I'm hoping we'll be able to have cells go from initial inoculation to carbon starvation in about 2 weeks or less than that. And we'll have to time our experiments based on what phase we want to perform our experimental procedures on. In terms of the grand scheme of things, it's a little difficult at the moment. I will admit this will be my first time doing any cultures, so I'm sure there's going to be a little bit of a learning gap on my end for proper technique. But I can't give you a timeline for when I'll have publications coming out.
Oh, yeah. And my second question is just kind of do with the I think you mentioned like a cobalt irradiator or something like that. Is that like the source of radiation? Is there any way you can check like that? Like if you plan for your culture to get like 100, I forget what the unit was, but 100 of whatever the dose of radiation is there a way you can check that it actually got that, or is there any concern with there might be like variability of the cobalt like laser or radiator itself or anything?
So we've had commercial companies come in and they performed what's called the dose map of the irradiator. So there's kind of a gradient where there's a small window, I think it's 4 inches in diameter and 6 inches tall, where we will receive 100% of the dose. And as we get closer to the walls of the container, that's going to increase from about 100 to, I think, upwards of 130. So as long as we're within that window, we do have some reassurances that we will be receiving the dose that we intend.
Okay, that's good. And that's all I had. Thank you for your presentation.
Thank you.
Any other questions from the audience? Let's see any hand raised up. I think I still have I'm gonna ask one more. It was like such an interesting topic for me personally. So I know you mentioned melanin being such an important pigment associated with it. Now melanin, as you also mentioned, humans have it as well, right? So is there a difference in the pigment itself? Why are we not capable of converting that ionizing radiation into energy or something?
I want to say that the reason why we may not be able to do it is largely comes down to a radiation biology question. And the more complex that the DNA is, the more opportunity there is for damage. So we may, from a totally, this is off the cuff of me talking, if we were to be able to harness enough energy for something like radiosynthesis to occur, would we receive enough biological damage to effectively kill us before we see a net gain? Whereas, for instance, fungi have much simpler DNA, and they also have much more robust repair mechanisms. So they may actually come out with a higher net benefit and magnitude than humans may ever be able to.
That's interesting.
Is there any? So, I'm sorry to jump in.
No, go ahead.
So we're talking about the energy generation. How about metabolizing oxygen versus radiosynthesis? I would also imagine for something in our size, maybe oxygen is much better, even if it's not as efficient. It has a lot more output of volume. Or maybe radiosynthesis is still better in that regard. It's just we are not as tough to radiation, like you said.
Could you repeat the question? I was having trouble hearing.
Okay, so I was thinking metabolizing oxygen, could that still be a better way to generate energy compared to radiosynthesis? Could that also be playing a role in terms of either efficiency or volume output?
So if we can find evidence to support radiosynthesis, it would be imperative that we kind of calculate the amount of energy that can be gained from that relative to traditional metabolic pathways. So for instance, we might have a spread of 95% aerobic respiration versus 5% of energy come from radiosynthesis. But if you think about it, back to what Rapunjali asked from an exobiology standpoint, especially before we had an oxygen-rich atmosphere, if that was the only means for energy to be metabolized, then that may be a little bit of a factor behind the slow exponential growth of life coming to as we know it.
Thank you.
Any more questions? Okay, if there are no more questions, let's all thank Steve for an amazing and a wonderful talk. We all really enjoyed it, Steve.
Thank you all for tuning in. I really appreciate it.
And good luck with your prelims, was it? I forgot what's the word.
Thank you. Thank
good
you
luck
so
for that too.
I'll need it.
Yeah. Bye, everyone.
Take care, everyone.
Bye. Goodbye.
Woo! I kick butt, yeah, yeah, yeah. I kick butt. Woo! Yes, I crushed it so. Oh. Huh. Huh. Man, that was fantastic. I feel like the albatross is off my neck.
Albatross.
Yeah. Yeah. Now I can hit the gym feeling like a million spondoolies.
Yeah.
And go from there. I had some issues too at the start of everything, where people weren't able to hear through my microphone, or I wasn't able to talk through my desktop microphone. So what I had to do is I had to snap my laptop open and then log in through my laptop while doing the presentation on my desktop.